on. Oh, there you go. Okay, this meeting is being recorded. Okay, so what we have to do is we have to very tightly regulate cell division, because when cell division gets out of control, that's basically cancer, right? Is when we cannot control cell growth. So because of that, cells have to be very regulated and when they can actually go through this entire process. And we regulate cell division via what's called the cell cycle. So the cell cycle is separated, as we've talked about before, into interphase, and then 2 stages of interphase, G1, S, and G2, which we're going to talk about in a minute. And then the actual nuclear and then cell division, which is mitosis and cytokinesis. We're going to talk really importantly about these 3 phases. What a first day. These 3 phases within interphase, G1, S, and G2. I think that's on our next slide. So we'll go back here. What I want to mention is G1 and G2, the G in those stands for growth. So what's happening in G1 and G2 is the cell is getting larger. So during G1, the reason a cell is getting larger is if you look at when G1 takes place, it takes place right after mitosis. So basically we have mitosis occur, we have 2 daughter cells, and these 2 daughter cells are relatively small, they're new, and you can even see a size difference between one of these new daughter cells and a cell within G1. So during G1, a cell is kind of like a baby cell, and it starts growing up a little bit to become an adult cell. After it gets out of G1, it enters what is called S phase. And S phase, the S stands for synthesis. And synthesis means to create, and in this case, what we're talking about is DNA synthesis, which is replication. So that's the same thing. DNA synthesis is the same as DNA replication. And you can see that's exactly what happens. A cell kind of grows up, goes from baby to adult in G1. And then once the cell enters S phase, what happens during S phase is the DNA is duplicated via DNA replication. All right. So G1 to S, this transition right here is when a cell goes from having basically 1 copy of each, or 2 copies of each chromosome, right? And then here it has 4 copies of each chromosome. So here we have our homologous chromosomes, and here we have our sister chromatids, and so our homologous pairs, just to remind you of that terminology. So after we have duplicated DNA, we need to grow a little bit larger, right? Because if we're going to separate this DNA, we need our cell to be large enough to separate. We also, during S phase, are replicating a lot of organelles. We're making more organelles, we're making more mitochondria, we're making more chloroplast in your plant cell, basically more organelles, so that when the cytoplasm divides, you have organelles in both of those daughter cells as well. So that's kind of what's happening when we go from S to G2. So we have duplicated DNA, and we're kind of duplicating everything else, as well as just generally getting larger. So that's S to G2. And then we have to go from G2 to actual mitosis. So that's basically during G2, we're large, we have 2 copies of our genetic information, and we are ready to divide. And then when we enter cell division, as you know, that's when we actually divide the cell. So as we will talk about, there are some critical stages here, but some of the most critical stages are those transitions from G1 to S, right? So whether we're going to duplicate DNA or not, or S to G2, which is whether we're going to prepare for cell division or not, and then G2 to M, which is mitosis, which is going from a cell that can divide or is prepared to divide to a cell that is actively dividing. So those are regulated by what are called growth factors. So you're looking at a very similar figure, we just don't have the plants here, but this is a depiction of the cell cycle. And you can see we have G1, which is where we're growing, but we only have one population of genetic information. We have S, where we duplicate that genetic information. We have G2, where we grow larger and prepare for cell division. And we have M, where we actually divide our genetic information. So how do we go from G1 to S, from S to G2, and from G2 to M? Basically, a cell has to have signals telling it that it can divide. So there's a lot of different signals that a cell can get. This is, remember when I think during, did we talk about this? We haven't talked about introns versus exons yet. But when we do talk about it, what we will talk about is that a large percentage of your DNA does not actually code for like genes that code for characteristics. So for example, some of your genes code for traits like eye color or hair color or height or whatever. Those are kind of physical manifestations of genes, or a gene coding for like a protein. Some segments of DNA do not actively code for things, right? So instead, they code for molecules that regulate things. So actually the vast majority, as we will learn, of your DNA codes for regulatory information. So we'll talk about that later when we talk about protein synthesis. But what I want you to know right now is that some of those genes code for what are called cyclins and cyclin-dependent kinases. So notice that those are not highlighted. That's because you don't need to know exactly what they are. I'm just throwing it out here so that you've basically heard those terms before. But cyclins and cyclin-dependent kinases, or CDKs, are what we call growth factors. So growth factors are just extracellular signals that tell a cell whether or not they are allowed to progress through the cell cycle. And you have a couple of different ones of these. I can't remember if I have this telling you exactly the CDKs. I do in a minute. But before I talk specifically about growth factors and transitioning basically stage to stage, I want to talk about another way, a really important way that a cell kind of gets a signal as to whether it can divide or not. So growth factors or extracellular signals are a really important one. Obviously, you can imagine when a sperm and an egg come together and form a zygote, what that has to happen is that zygote has to grow and divide into an organism, right? We have to go from one cell to like lots of cells, like what we are right now as organisms. So in order to tell the cell, the zygote, to do that, you have to have extracellular signals. And those extracellular signals are called growth factors. So we'll talk about those in a minute. Another way that a cell can decide whether it needs to grow or not is called contact sensitivity. So I will add a little mark here because that's where I'm talking about it. Oops. All right, so contact sensitivity is what we're looking at when we look at that picture to the left, so over there. So basically what you're looking at here on the left in this figure is a petri dish, sort of, just a dish. And you can see that we have a couple of cells in that dish. So cells, they like to be kind of anchored to something. They have to be anchored to something. So in class, we've talked a lot about like the skin. And the skin is going to be a perfect example for contact sensitivity as well. So obviously you are aware that when you grab your arm and you twist, your skin is not flying around your arm. So we talked about the fact that your cells are anchored to connective tissue, which is under them, right? So we talked about that. And another thing about that is with that anchoring, cells will not grow unless they are anchored. So for example, we talked about the stem cells in your skin anchored to the connective tissue, and it's those stem cells that, because of that anchoring to the underlying connective tissue, they are given the signal to grow out, and they produce all the outer layers of your skin. All right. So what happens with anchorage is you have a cell anchor to underlying connective tissue, and then that cell starts to grow. So you can see that you have anchorage, and then in the next figure down, you have those cells starting to grow. We go from a few to more. All right. And what starts to happen is, notice that the cells only grow until they contact each other, right? So notice it says a single layer of cells. That's because cells grew from just a couple, and they grew until they touched each other. So what will happen is that your cells will grow until they touch each other, and then that gives them a signal to stop growing. So this is really, really important. So what I want you to think about, right, I'm going to turn on my video for this because it's a little bit easier for me to kind of explain that way. All right, let me find myself. All right, here I am. Okay, so it's a little bit easier for, dang it, what's going on here? Whatever. So think about if you have your arm, right? And if you get a cut in your arm. Anytime you get a cut in your arm, you'll notice, or a cut in your skin, whatever, you do get scar tissue, which tends to be a little bit kind of outgrown on your skin. But what happens is if you get a cut in your skin, those cells basically grow until they contact each other. So you go from a cut and your skin grows until it contacts the skin on the other side. And then really importantly, because it has contacted the other side of the skin, it stops growing. That's called contact sensitivity. So think about it this way. The reason this is so important is for that same reason, is if you had a cut in your skin, right? So you go from a cut and that skin starts to grow together. What would happen if that didn't stop growing? You would get a big mound of cells that would continue growing and growing and growing and growing. Correct? Yeah. So if you get a mound of cells that keeps growing and growing and growing and doesn't have the signal to stop, does anybody know what that's called? You can type it in the chat. Yeah, good job. Cancer, tumors, right? So contact sensitivity is one of the most important kind of ways that a cell, if it encounters another cell, it won't grow anymore. So that's where you go from skin, you go from a cut, that cut grows until it contacts the skin on the other side, and then it stops growing. If it doesn't stop growing, you get a tumor, right? Which is cancer. So that is one way that cells can, that is one way that cells can basically stop growth or regulate the cell cycle. Basically what they would do is if they're in G2 and then they contact another cell nearby, they would not enter M phase. That would stop them at, you can see in this figure, the G2 checkpoint would stop them from dividing. Okay, can I do another survey in the chat? How many of you feel better seeing my face? Or just don't care? I can explain with my hands. Okay, okay, great. Well, I'm just gonna leave this. I'll just continue to put makeup on for you guys. I'm all about it. Okay. As for split screen, I think that you have to do that on your end. So if you want to split the screen, you can kind of go and manage that in your settings. All right. And I appreciate that. I probably won't wear a lot of makeup. So here we go. So we understand how that cell cycle can be regulated. So we know that we can kind of go through the, thank you, Erin. So the view options side-by-side mode. Yeah. Do I need to do that on my side? Or I'm pretty sure that's everybody's. Let's see. Awesome, good, awesome. You guys are so good. I love our little virtual classroom. Okay, all right. So we're talking about regulating whether we go through cell division or not. So again, one way is that if a cell contacts a cell next to it, it won't continue growing. However, we also have to talk about how cells can grow that from other external signals. So that's what we're talking about when we talk about checkpoints. So checkpoints, you can see on this figure, are basically where we go from G1 to S. You can see that's called the G1 checkpoint. Or if you go from G2 to M, that is called the G2 checkpoint. And we actually have a checkpoint in the middle of M. So basically, if a cell is actively dividing, and then we can actually tell a cell that's actively dividing to stop, right? And so that's called the M checkpoint. And then you'll also notice something during G1, this little arrow that pops out, that's called G0. So this is where a cell gets stuck in a phase, kind of, where basically it's told to not even go into cell division. So it's basically just a cell, and it is not going to divide. So let's talk about each of those checkpoints. So first off, the G1 checkpoint, as the PowerPoint slide says, basically, if the G1 checkpoint, I get a little frustrated with some of the terminology, but sometimes you will hear that the G1 checkpoint is like the most important checkpoint of the cell cycle. I don't think that any checkpoint is any more important than any other checkpoint, for the simple fact that all of these checkpoints are regulating cell division, which is very important. However, a lot of kind of textbooks and other will say that the G1 checkpoint is the most important checkpoint. The reason they say this is because typically if a cell passes through the G1 checkpoint, it's basically going to go through mitosis. Even though we still have the G2 checkpoint, we still have the M checkpoint, if a cell gets through the G1 checkpoint, it's very likely that it's going to divide. So if a cell is in G1, so basically, again, we go from one cell to two cells, and each of those cells are in G1, so growing, going from baby to adult. When you're in G1, you have two options. One, you can go through the G1 checkpoint, in which case you'll probably go through mitosis and make more cells. Or you can enter G0, which is basically a cell saying, I'm an adult and I'm not going to divide. So there are a lot of cells in your body that do this, enter G0. Muscle cells, nerve cells are a really important one. So several of you probably have heard, and we might have talked about this in class, I can't remember, but one of the reasons nerve damage and neurological disease and neurological damage is so bad and so severe is because nerve cells are in G0. So what that means is if you have a nerve cell, it is in the state it's going to remain in through its entire life. And if that cell is in G0, it's not ever going to divide to create new nerve cells. So what this means is that our body has a really hard time generating new nerve cells because the vast majority of nerve cells are in G0. So that's why it's really difficult to get kind of whenever you have neurological damage, your body doesn't necessarily have the cells that are capable of repairing the loss of that nervous tissue. Muscles are the same, which is why, muscles are not the same, There are muscles that, or muscle tissue that is not necessarily in G0, but one of the reasons, one stimulus for muscle growth is like exercise and weightlifting and kind of using your muscles. That would be an external stimulus that tells your muscles to kind of create more muscle tissue. So one thing that can happen is if a person is put on like bed rest, or if they don't have the capability to use their muscles, they won't be stimulating the creation of more muscle cells. And this is why muscle degeneration can happen so rapidly, especially with like bed rest or things like that. And it also explains why it is hard for somebody who's coming off bed rest or injury, why they have to go through like PT or OT to kind of get their strength back, and why that takes so long. It's because the vast majority of your muscle cells are actually in G0. It takes a really long time to regenerate muscle, a lot of effort to regenerate muscle. All right. Okay, so that's G1, again, sort of one of the most important checkpoints. If a cell gets through the G1 checkpoint, it's probably going to go through cell division, or it has the capability of going through cell division. Cells that are in G0 will not be moving through the G1 checkpoint, and therefore they're probably not going to divide. So that's nerve cells and muscle cells. If a cell gets through the G1 checkpoint, it enters S phase, and S phase is when it duplicates that DNA, the genetic information. After it duplicates the genetic information, it starts to create all the other organelles. Kind of if you're going to have that much genetic information, you also need to have the resources to support it, which is the purpose of G2. So you'll notice there is not a checkpoint between S and G2. That is for that reason. If you have duplicated DNA, you need to have the resources, which is the cytoplasm and the organelles to support it. So if you go through S phase, you're going to go through G2. There's another reason that if a cell gets through G1, it's probably going to go through mitosis. Because if you go through G1, you're going to make duplicated DNA, and if you have duplicated DNA, there's no reason to not go through cell division. All right. So then a cell has to go through G2, or the G2 checkpoint, in order to get to cell division. All right, sorry, gross, as always. So a cell has to get through G2 to enter mitosis. The G2 checkpoint is basically what is passed through hand sanitizer, y'all. All right. This was a gift from a student. I didn't realize how precious it was at the time. Okay, so G2 to M, you're going to go through mitosis. And then, again, a cell starts dividing, and you'll notice that the M checkpoint is basically occurs after the cell has divided the nuclear information, the DNA, and then the next step, like we've talked a lot about, the step after dividing the actual DNA is to divide the cytoplasm. So that's what the M checkpoint allows to happen. So basically, if you go through the G2 checkpoint, you've divided the DNA, and if you get through the M checkpoint, you're dividing the cytoplasm. All right, so those checkpoints are heavily, heavily, heavily regulated. Again, they are regulated, if we go back, they are regulated by growth factors. So there are growth factors or molecules that allow us to get through the G1. Growth factors are molecules that allow us to get through G2 and M as well. Primarily, these are going to be those cyclins and cyclin-dependent kinases, but there are others. And this can also include like contact sensitivity. And importantly, if this is not tightly regulated, you get disease. And as you all put forth, the disease we're talking about is cancer. All right, so speaking of cancer, well, before we go into this, are there any questions? Everybody feel okay? All right. All right, good. So just a reminder that if you have questions, put them in the chat, but I will also have a discussion board after this, and yeah, we'll be good. All right. Okay, so if there's no questions, let's go on. So if we're not regulating the cell cycle, if the cell cycle is not regulated, oh, we have a question. All right, so is there no checkpoint in S phase to reduce the chance of mutation in the DNA? This is an excellent, excellent question. So notice that we have a G1 checkpoint that regulates whether we go to G1 to S, and then after that, we don't have a checkpoint until we go from G2 to mitosis. So the question kind of is pointing to, what if during DNA synthesis, if we have a mutation, right? Because if we have a mutation, we don't necessarily want to enter mitosis. And the answer is I guess the answer is a little complex. So first, let me revisit this slide. I had it, and then this slide, to remind you that your cells do make mistakes. And one mistake that can happen during S phase is kind of defects that would lead to improper cell division. Non-disjunction is a defect in mitosis, but what can sometimes happen with that is that the mutations in a cell that allow non-disjunction to happen can actually happen during cell division. So that's a little bit complicated. We're not going to get into that. But what I will say is that that should point out that if there are mutations during S phase, there's two possibilities. One, the cell will still divide, and you'll get those mutations passed to offspring, and that's what happens kind of with non-disjunction. Or if there is a mutation or something devastating that happens during S phase, the cell will actually not progress, but it won't just stop, it'll just die. So that is via a process, and I'll type it in the chat box, called apoptosis, sometimes pronounced apoptosis. I say apoptosis, actually I say both, I guess. But basically what apoptosis is equals programmed cell death. Hold on. I am everyone. Apoptosis is programmed cell death. And basically what that is if a cell detects some sort of error, it can say, I can't survive any longer. So that would be stopping mitosis, it just wouldn't be a checkpoint. I hope that makes sense. Feel good? That's an excellent question. Okay, so yeah. Okay, so if we do have a defect or something like a cell is getting through a checkpoint when it shouldn't be, and we're getting cell division when we shouldn't be, that is cancer. So we have some terms for cancer, and these are really important. First off, we classify any kind of mass of cells that are growing because of uncontrolled cell division. That is a tumor, right? So anytime you have a bunch of cells that have grown out of control, so basically those cells have bypassed those checkpoints, G1, the G1, G2 checkpoints. What am I missing? G1, G2, or M checkpoints. That is a tumor. It's uncontrollably dividing cells. Sometimes this is bad, sometimes this is okay. And that's basically how we classify these tumors. So again, a tumor is just a mass of uncontrollably dividing cells. You can have what's called a benign tumor, and a benign tumor means that that tumor is relatively safe. The reason it's relatively safe is because it stays at the original site. So for example, we talked about like if I cut my skin and I had the cells grow right here, if that tumor or that mass of cells stayed on my skin, that would be a benign tumor. It would stay there, right? What can happen though is because all of your entire body and all of an organism is very intimately connected by the bloodstream as well as the lymphatic system and all the other systems in your body, is sometimes if you have a cell that is continuously dividing, one of those cells can pop into the bloodstream or into the lymphatic system and travel somewhere else. In this case, that cell would kind of travel to a different location and it would like stay in a different location. So let's say like one of those uncontrollably dividing cells from my skin, from that tumor on my skin, entered my lymphatic system and traveled to my lungs. Okay, that cell is still uncontrollably dividing. So it sticks in my lungs and then it starts uncontrollably dividing there. So remember, one of our core concepts in this class is that structure equals function. The structure of my lungs is extremely important to their function. They have to expand and contract. They have to be in their proper structure to be able to do that. Yeah, exactly. So is that called metastasis? Yes. So if we have one cell pop into either the circulatory system or the lymphatic system and go to another site, that is called metastasis. So basically metastasis is movement of a tumor cell. So what would happen is if that cell starts proliferating in that area, right, it's not supposed to be there, and all of a sudden my lungs, which are basically just sacs, have this big old ball of cells there, that's interrupting structure, which interrupts function. It makes it hard to breathe, right? So if a cell is capable of metastasis, so if a tumor cell is capable of moving to a different type of the body, that's what you would call a malignant tumor. So if a skin cell, got into my lungs and then started dividing in my lungs, that cell would be a malignant tumor, right? It's benign if it's a skin cell growing on my skin and not capable of moving. It's malignant if it's a skin cell that has moved to my lungs and it's growing and interrupting function. Again, like if it's interrupting function, that's malignant. And that's also cancer. So cancer specifically is a malignant tumor, right? So not all tumors are cancer, but all cancer are tumors. It's one of those crazy mind puzzles. Okay, so again, a tumor is just uncontrollably dividing cells. You can have benign tumors, which are somewhat less dangerous because they tend to stick in the given location. They don't necessarily have the capability of moving. A malignant tumor is one that is capable of moving and therefore would interrupt structure and function. So you can see on the PowerPoint slide that one of the really concerning type of cancer is breast cancer, and that's what this is showing. This is showing breast cancer. One of the reasons breast cancer is so concerning is you can see on the left, you can see the tumor shown in red. You'll notice that breast tissue, you see the glandular tissue. Glandular tissue is a tissue that secretes stuff, so for example, milk, but because it secretes stuff, it has to get a ready supply of fluid, and that fluid is supplied by the circulatory system and the lymphatic system. So glandular tissue, anytime you get a tumor around glandular tissue, that can be sort of concerning because location-wise, it is very close to circulatory and lymphatic tissue. So you can see here in this breast tissue, you have the tumor growing around glandular tissue. That tumor grows right now. That would be concern. How fast do tumors begin to grow? Excellent question. The answer for how fast do tumors grow completely depends on the type of tissue that has a tumor in it. So for example, glandular tissue is very proliferative. So breast cancer tumors tend to grow relatively fast. Another type of tumor that has the potential to grow really fast is pancreatic tumors. Our pancreatic cancer is very serious because it has a very quick progression. The pancreas is glandular. It secretes stuff. It secretes insulin. It secretes Glucagon, it secretes those hormones that are important for kind of regulating that. And so because of that, it grows very fast. Pancreatic tissue, breast cancer, all of these are kind of very proliferative tissues. They can grow so fast, and one of the reasons they're so concerning is because they can progress so quickly that it's hard to detect them early. And for any of these, if we want to stop tumor growth or stop cancer growth, early detection is key. Another question, excellent question. Is it true that most everyone has some sort of benign tumors in their body somewhere, but most don't become malignant? Which is why it's recommended that everybody get a baseline CT scans as normal health screening. That is an excellent question. It's a little complicated. I can't give medical advice, even though I'm a doctor and my husband's a physician, which I remind him of daily. Just kidding. Except for right now, I'm like, tell me what to do. But he's never been more useful. But with this, I would say that I don't think everybody has benign tumors in their body. So I don't think we are all hanging out with benign tumors. Because our cells are always proliferating, so growing and dividing, we always have dividing cells in our body. So that is absolutely true. Not all of these cells will be dysregulated and turn into tumors. However, as for whether you can detect whether a cell will become malignant if it's benign, that's pretty hard to do. So what I would guess is that if your doctor detected a malignant tumor, they're going to suggest it be removed. So if it's easy to remove, so a lot of times with like skin cancer, they just remove it, or they will biopsy it, which is where they take a little sample of it, and they look for cells that will be capable of moving, basically. It's a little bit difficult to explain what would allow a cell to move, except to say that your cells are designed to be in the tissue they're in. So we've kind of talked about this, where like the cells on my skin surface are dry, basically dead, filled with keratin. So those cells, in order to function, have to be dry and filled with keratin, etc. Whereas the cells in my lungs have to be moist, they need to have a lot of organelles, they need to be able to secrete what's called a surfactant, which is to keep my lung tissue moist so it can expand and contract. If you put one of these cells in my lungs, that'll interrupt function, but also it actually wouldn't survive normally. If you put a lung cell on my skin surface, same thing, like it wouldn't survive. So another thing that would allow a cell to metastasize is that it basically has to be able to grow uncontrollably, but also it has to be able to sort of survive independently of its setting. So when a doctor is able to do like a biopsy or look at a tissue sample, they're not only looking like, is this constantly dividing, but they're also looking at would this cell have the capability of surviving elsewhere in the body, if that makes sense. So although like CT scans would be probably pretty good, right? Telling you like, oh, do you have any tumors? Do you have any irregular tissues? That's sort of like, I would say like with normal health screenings, it would depend on how regular those are. And I would say that that would probably only need to be part of a normal health screening if you have risk factors in place. So for example, like people with the BRCA1 gene, which kind of predisposes you to breast cancer, absolutely. You're gonna need to have more kind of increased CT scans. The problem with any sort of radiology is that there are risks involved, right? So exposure to stuff that could cause mutations and that could cause sort of tumors. So doctors should be very careful about how often they require that, if that makes sense. Yeah, that's an awesome question. Good question. All right. So the other question, would endometriosis technically be considered a cancer, especially if it's one where the endometrial tissue is growing other places in the body? That's another excellent question. Endometriosis is so crazy, and I do not believe that it is characterized as a cancer because it can't necessarily spread to other areas of the body. What happens with endometriosis is the lining of the uterus starts growing outside of the uterus. So it's still associated with the uterus. So it starts to interrupt function for sure, but I do not believe that that endometrial tissue can kind of enter the lymphatic or the circulatory system and spread throughout the body. And it's not necessarily uncontrolled cell growth so much as it is cell growth in the wrong location, if that makes sense. So it's a little bit different, but there are characteristics that are similar. That's a really good point. Excellent question. Thank you. All right, so excellent questions. All right, so tumor, uncontrollably dividing cells. If it's benign, it's going to stay in its place, but still grow. If it's malignant, it has the potential to spread. All right. Another thing I want to mention is that if you have a cell that is a tumor cell, and it basically just like cannot be controlled by the cell cycle anymore. So it can basically fly through G1, G2, and S or M checkpoints without issues. These cells are called immortal cells. So again, not only have these cells lost the ability to kind of stop the cell cycle, so they're always going to be going through cell division, but immortal cells also lose contact sensitivity, so they're not going to stop growing if they contact each other. And also these cells have the ability to kind of grow anywhere. So immortal cells are actually, when we use that terminology, we typically use that terminology in a lab setting. And we actually can use cells that are immortal as what we call cell culture in a lab. So if we kind of go back to this slide, if you look on the left, this picture, what you're looking at in that slide is cells that are basically put on a plastic dish, and then they start dividing. Normal cells in your body, if you take them out of your body and put them on a plastic dish, they're going to die. They're going to be like, I'm not supposed to be here. What's going on? I'm not getting signals. You know, however, immortal cells, because they have lost cell cycle control, and because they have lost the necessity for external factors controlling them, you can basically take those cells, put them on a plastic dish, and they will divide and they will grow. So what you're looking at on the left in this picture is a collection of immortal cells. The reason that matters is because if any time in a lab you want to test the safety of a drug or a chemical, or if you want to look at biological processes, it's not really ethical to like do that on a human being, right? So right now in Seattle, they're actually testing the phase 1 vaccine for COVID-19. Very exciting. A bunch of my friends are like, yay, phase 1. And then a lot of people are like, how do I sign up? I would hesitate to recommend anyone to sign up for a phase 1 clinical trial, because a phase 1 clinical trial will only come after basically they take immortal cells in a petri dish and test whether the components in that vaccine have any effect on those cells. So that's going to be like, do they secrete, you know, immune proteins that would indicate some sort of reaction to that chemical, or are they killed by the vaccine, etc., right? So phase 1 clinical trials occur after a significant amount of time looking at the effect of those chemicals on just immortal cells. Sometimes primary cells, which are cells that are extracted from a human, those are going to be like stem cells that we can use. but a lot of the times that data comes from immortal cells. So there's issues with this. Like we talked about the fact that immortal cells are sort of not regular cells. They can survive in pretty harsh environments. So people that enroll in phase 1 clinical trials are actually, that's a pretty risky scenario because it's the first time that a chemical, a drug, or a vaccine or whatever has been used on like a living, breathing human. And so I am very happy for the people that are volunteering to be a part of the phase 1 clinical trials for COVID-19. Would I volunteer to do that myself? Probably not. But I'm glad that people do volunteer for it. I will say that they wouldn't allow people to volunteer for it unless there was like tons and tons and tons of data supporting that it was safe at that time. So immortal cells are basically taken from a tumor, wherever that comes from, and then we can just like perpetuate them and grow them in a lab. One of the most famous examples of an immortal cell used in lab is HeLa cells, which I have as the example down here. HeLa cells are named after the person, Henrietta Lacks, and she had cervical cancer, and basically the cells from her cervical cancer were extracted and they've been used in the lab ever since, right? So that was like 60 or 65 years ago at this point. You can read an entire book about that called The Immortal Life. I'll type it up here. Oops, yeah. Of Henrietta Lacks, which is an excellent, excellent, excellent book. And it talks about how that cell line was derived. It also talks about Henrietta Lacks herself and her family and the struggle to have, unfortunately, the struggle that they have faced, like having credit for that person's cells and how those cells have been used. And I mean, if you don't feel like reading a book, I'm pretty sure that it was turned into a movie with Oprah. So I get it. Today on NPR, the New York Times, they were like, here are 9 shows to binge during the pandemic. And somebody commented like, oh my God, why don't you read a book? And I'm like, because I like to watch TV. I like movies, so leave me alone. And I actually did comment that because, as I mentioned, I'm a huge troll online. I'm just kidding. I am. Okay, so, all right. So immortal cells are cells that are not controlled by the cell cycle. They also are not kind of affected by extracellular signals. Another thing that I want to talk about, I don't know why I have it a bullet under that, but chemotherapy. So if a person has cancer or has a tumor or anything like that, they're often put on chemotherapy. Chemotherapy drugs are designed to hopefully stop the cell cycle from progressing. So basically what it does is chemotherapy drugs, I apologize. What chemotherapy drugs do is they try to take these cells that are replicating or dividing out of control, and they try to stop that process. So, yeah. All right. Do we feel good? Any questions about cancer, immortal cells, or chemotherapy? Please don't touch your face as much as I am touching my face. Feel good? Any questions? I don't see anything. All right, I appreciate it. All right, so let's go forward. Actually, what I'm going to say is if we go forward, we're going to be entering a whole different unit. So we're going to be not a whole different unit. That was horrible. We're going to be entering a whole different topic, which is inheritance. So inheritance is awesome. It's awesome. However, we only have like 15 minutes left. So I think what we're going to do, I want to introduce inheritance and what we're going to be talking about, but we'll probably talk about the bulk of this on Thursday. But just to like kind of introduce you to what we'll be talking about on Thursday, since we do have a couple of more minutes, I do want to mention that we haven't talked about this yet. We will be talking about this later. I believe we talk about this in Unit 3, so after our next exam. But in the future, we will be talking about one of my favorite and arguably one of the most important concepts in biology, which is protein synthesis. And protein synthesis is when your body takes its DNA, the genes in your DNA, and sends it through two processes, transcription and translation, in order to take your genes and turn them into whatever they're going to be. So this is typically protein, sometimes it's RNA, but basically when we talk about protein synthesis, what we're talking about is going from a genotype, so the genes you have, to a phenotype, which is the expression of those genes. So for example, on this slide, you can see a genotype is a gene. So in this case, your genes are made up of nucleotides like adenine, thymine, cytosine, and guanine. So I have A, T, C, G, C, C, G, blah, 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 blah. That's a genotype, just the nucleotide sequence that you have. And then when we read that through transcription and translation, that's read into a phenotype, which is the physical manifestation of that genotype. Again, this is typically due to proteins, genes becoming proteins, which is why this is called protein synthesis, or gene expression is another term which means the exact same thing. But you can think of a genotype as the sequence, your genetic sequence, your gene sequences, and a phenotype as the outward expression of that. So if your genotype was ATCG, whatever, and your phenotype is, I have a box around like green eyes, right? So the green eyes, the physical manifestation of the genes you have, your eye color would be your phenotype, right? So another way you can say that is the genotype is the DNA and the phenotype would be the protein, or what we actually see as a result of your genes. So this is really important for inheritance because what we're going to talk about is we will talk about genotype and phenotype, because what you inherit are genes. What is passed down in DNA is genes, but right, the information you inherit, if your mom has blue eyes and your dad has brown eyes, how do you end up with like green eyes? All right, just to make sure, phenotype is DNA, right? So I'm going to type it in here, and I will also reiterate this later. DNA is your genotype, and protein, or the physical manifestation of your genotype, equals your phenotype, what you observe. Good. All right. Actually, what I'm going to do is right. There you go. All right. There you go. Hopefully that should help. We'll talk more about this as we go forward as well. Okay, so again, what you inherit, if your dad has brown eyes and your mom has blue eyes, how can you have green eyes? It's because you're not inheriting a phenotype, right? You're not inheriting blue eyes or brown eyes. You're inheriting genes that lead to those phenotypes. And those genes, when mixed up, may not lead to either of those. It may lead to like an in-between or something. So that's what we call inheritance, and that's what we're going to be talking about moving forward. We're going to be focusing on genotype and phenotype. So what I'd like to do before we start talking about this is kind of we will be talking about Mendelian genetics, which I remember talking about as a student. Again, you all know this, I am a medical microbiologist. I am like a medical biologist. I am very medical oriented. And so if you're going to talk to me about plants, I sometimes shut down. I'm like, okay, I don't care, which is unfortunate because they're very important. However, we will be talking about Mendel, we will be talking about the pea plants. because it is very, very important and demonstrates these important points well. I think it's important to emphasize why that those experiments were so important. And a way to do that is to kind of try to explain, like, before Mendel came along, what we actually thought, or how we actually thought stuff was inherited. So this was all before the 20th century. Mendel was a monk, and he had a lot of time alone, and he was a wonderful botanist and had a lot of time to sort of be tedious with his experiments, very tedious with the experimental scientific method, which led to literally the basis for our modern day understanding of genetics. It's still all very relevant. But before Mendel did that, basically there were two major hypotheses as to how we inherited traits. And I giggle a lot because I think these are outrageous, but it makes sense that people believed it. So before Mendel came along, one of the kind of first major hypotheses was that traits were acquired via the technical term of blending of fluids. I cannot even say it out loud. But basically what I thought happened is that fluids from the mother and fluids from the father were just like blended, and somehow those fluids would, you know, like combine into a baby. So if you haven't heard of like humors before this time, a long time ago, before we understood like organ systems and how the body anatomy and physiology, we believed that there was sort of like humors. And that was kind of the blending of fluids was sort of like the blending of humors. And basically like mother and father fluids were blended into like a new fluid. So, but the reason that didn't make sense is because obviously we are not an equivalent blend of our parents. Some of us will have like the exact eye color of our mother, or the exact, like, you know, just traits will be so similar to our mother or father. It's not a blend of the two, it's like one or the other. So there's one that was not true, obviously. The second one, which I think is hysterical and it is so characteristic of the time, is that it was believed, as shown in this picture on the slide, that every little sperm had a tiny baby in it, and that when the sperm entered the mother, that baby was like planted in the mother, and that's where it grew. So obviously this is like way off, because babies have characteristics that are inherited from the mother. It's not just the father. However, this is very, like, explains a lot of the societal kind of, back then it was like illegal to masturbate, and it was like illegal for extramarital sex, or sex that shouldn't directly lead to reproduction. This is why. It's because, like, if sperm was released, you're killing babies. So obviously we know that's not true. So, you know, Mendel really had made some very, very important discoveries that are literally the foundation for our understanding of modern day genetics. So I like to point that out because it is extremely important for us to understand that without Mendel being super tedious and also super isolated, we wouldn't have modern day genetics, all right? So we are all isolated in this pandemic, but some of the most important discoveries, genetics, right, came from isolation. All right, so I expect you all to, you know, find the next foundational biological concept while you have a little bit of free time. All right, yes, exactly. Every service. Thank you, good. So, man, I need to watch that. I have time now, so okay. All right, so we will on Thursday pick up. We're going to talk about Mendelian genetics. We will have, we will talk about it in the context of his experiments, which involved pea plants, etc. But again, extremely foundational biological concept. So with that, let me see. Just a second, give me a second here. Let me come back to, I have a whiteboard. Okay. I just want to get back to the Zoom screen. I don't know how I can. Hold on just a second if you don't mind. Thank you for understanding while I figure everything out. Oh, there it is. Okay, all right. So again, this lecture, I will probably, after we get through the next couple of lectures, upload the modified version since I'm inserting text boxes, etc. I have recorded this lecture and I will upload that recording to either Canvas or I'll have a Google Drive where you can click on it and I'll have all the files on there, depending on how much space I have on Canvas. We will have a discussion board, probably at the end of the week, to discuss what we talked about this week, for any questions, etc. And yeah, so we have about 5 minutes left. Are there any questions about what we talked about today or how we're going to go through lecture or anything like that? Okay, so just to confirm the lab doesn't have a Zoom, that is correct. So this week's lab, what you need to do for this week's lab is you will go to your lab Canvas to take the lab quiz. It requires Respondus Lockdown Browser. So hopefully you've downloaded it. In order to access the quiz, if you've downloaded the browser, all you need to do is click on the quiz and it'll open the browser for you. And you have, starting yesterday at 11:45, and then you have until 9 p.m. today to take that quiz. You also have, I believe, until either today or tomorrow. I don't want to give you inaccurate information, but you have to, after you do the quiz, you need to go to our lecture Canvas and go to the module for virtual labs and go through their Connect virtual lab simulations for enzymes. So sort of what we were going to do in lab this week, a little bit different. In the future, and hopefully you'll see it populate soon in your lab sections, you'll be seeing a bunch of like Labster stuff, and that's not all due at once. It'll be a week by week thing. Just ignore it until I tell you not to ignore it, basically. All right. How will I get the recording for this lesson? I will be posting it on Canvas. I'll send you an announcement to tell you how you can get the recording. It will either be posted as a direct like file on Canvas, or I may make a Google Drive where you'll click on the link in our Canvas shell and it'll take you directly to it. So you will have access through Canvas and I will send you directions of how I do it once I figure it out. Your second exam will still be multiple choice, yes. I am pretty sure. Let me figure that out. Probably multiple choice, unless there's some like fill in the blank. I don't really think so. But it's all gonna be via Canvas. I'll give you more information about that as I get it. Future labs will not have Zoom, so I'll be virtual lab simulations. I'll send you more information about that. How are the exams going to work? They will be on Canvas. It'll basically be like a Canvas quiz, sort of like the lab quizzes, and I'll send you directions for that. You'll need the Respondus monitor. It'll be randomized questions, probably mostly multiple choice. There may be some fill in the blank, but I'll give you directions for that as we get closer. Clicker questions, we can't do clicker questions because of this format. What we're going to do in lieu of clicker questions is we're going to have a discussion board. So that'll be 1 per week, and you'll be getting points for participating in the discussion board. So I'll set that up today, but that's basically in lieu of clicker questions. I will still have clicker questions embedded in the lectures I give here, just so you can like see some questions and so we can kind of engage that way. But just because we don't have the clickers themselves, we won't be able to like get points for them. But you'll still see clicker questions and see the answers to them, and those will be very helpful for studying for exams as well. Will exams be curved, and will we have makeup bonus points? So yeah, because if a lot of you, what I will say, I don't know whether exams will be curved, and I need to figure out the bonus points. What I will promise you to do is that after we take our first exam online, is I will look at the basically how well you do and determine whether we need a curve or whether we need to do the bonus, right? So that information will be forthcoming. So I do want to point out though that I am very aware of the fact that this is crazy circumstances, and I'm being asked to work. I'm not allowed to be on campus after today. They're moving to essential personnel only. My daughter can't go to school, and so sometimes she'll be like, you know, behind me, screaming or watching Frozen 2, hopefully. But I am 100% sympathetic to the fact that this is, you are all in a very unique circumstance. Some of us may have it easier than others, and I am aware of that. And my goal is that moving online, we can still have fun. Like, I had fun doing this lecture today. I loved it. I loved interacting with everybody. I'm glad we're doing this. I'm glad we're doing chat. I'm glad we're going to do discussion board. I loved it. I want to keep our thank you. I want to keep our classroom environment as interactive and as good, and I want you to have as positive and a good of an experience in this class as if nothing had changed. I don't want your circumstances because of the pandemic to have an effect on your ability to do well in this class. So there's a lot of ways, right? The biggest thing is if you are struggling in any way, the best thing to do is to contact me and just kind of talk to me about your circumstances. I do ask you to please put in as much effort as you are able to do in finishing this class and making sure it's effective. However, you know, just keep me in the loop, right? So yeah, just keep me in the loop. I will do my best to support you so long as you are doing your best that you can in light of the circumstances to do well in the class. So just keep me in the loop. Again, we'll have the exam. It is scheduled. We're going to keep it as scheduled for the 31st, unless things change. It'll probably be sort of a thing where you have like 2 days to do it, sort of like the lab quizzes, 2 days to do it, sort of thing. But I will look at your scores, and based on your scores, I will for sure say, you know, ooh, we need to think about this, we need to think about this, right? So we'll talk about that. I'll figure that out as we go. It's going to be a fun learning experience. I'm glad we're in this together. Good question. Any other questions? All right. So I will send you a message out telling you where you can access the link to this lecture. You can also get this lecture closed captioned via Zoom, so that's awesome. I'll send out how to see the closed captioning, and look for a post about discussion board. Look for a post about how you can access the recording, and, you know, send me emails, communicate with me, talk to me. I will be having open office hours, meaning just like this, today from 11:30 to 12:30. So I will send out the link. It's actually the exact same link we have right now. So just between 11:30 and 12:30, if you want to talk to me, you know, click that link. I'm here. Good? All right. If that's the case, have a wonderful day, right? Get out, wash your hands, get a little vitamin D. You guys are the best. We're going to do this. All right. Bye, guys. See you soon.